In a basement laboratory in Cambridge, England, in 1977, Frederick Sanger worked with a tray of glass plates coated in gel. Each plate carried 4 narrow lanes of dark bands. Reading those bands from bottom to top spelled out the order of chemical letters in a piece of DNA. It was slow, and it worked. The method spread. By the late 1980s, a well-funded laboratory could read a few thousand letters in a day, and a single human gene might run to tens of thousands. The human genome, everybody agreed, contained roughly 3 billion. Nobody had a machine capable of finishing that in a human lifetime. The question hanging over biology was simple to ask and enormous to answer. What is actually written there? Not one gene at a time, pulled out because somebody suspected it caused a disease, but the whole sequence, start to finish, every chromosome, in order, available to anyone. The Human Genome Project formally launched in October 1990. It was a public consortium, funded chiefly by the United States National Institutes of Health and the Department of Energy, with major partners in the United Kingdom, Japan, France, Germany and China. The planned duration was 15 years. The planned budget was $3 billion. Its first director was James Watson, who with Francis Crick had described the double helix in 1953. Watson insisted that a fraction of the budget go to studying the ethical, legal and social consequences of the work. He opposed patenting raw human gene sequences, and resigned in 1992 after clashing with his superiors. Francis Collins took over in 1993. He was a physician and geneticist who had helped find the gene behind cystic fibrosis, a hunt that took years of painstaking work across a single stretch of one chromosome. He understood exactly how expensive the old way of doing things was. The consortium chose a deliberate strategy. First, build maps of each chromosome, dividing it into large ordered pieces cloned into bacteria. Then sequence those pieces one by one, knowing precisely where each fragment came from. It was methodical, verifiable, and it would take a very long time. In February 1996, the laboratory heads met in Bermuda and agreed on something unusual. Any sequence longer than 1,000 letters would be released to a public database within 24 hours of assembly. No embargo, no waiting for publication. The Bermuda Principles made the human genome, in effect, unpatentable as raw data. Meanwhile, the machines were changing. Fluorescent dyes replaced radioactive labels. Slab gels gave way to thin capillaries, and by the late 1990s a single instrument could run 96 samples at once, unattended, through the night. Cost per letter fell by orders of magnitude while the project was still running. In May 1998, Craig Venter announced he was going to do it himself. Venter had already sequenced the first free-living organism, the bacterium Haemophilus influenza, in 1995. His new company, later named Celera Genomics, said it would finish the human genome in 3 years for a fraction of the public cost. His method was whole genome shotgun sequencing. Rather than mapping first, you shatter the entire genome into millions of random fragments, read the ends of all of them, and let a computer find the overlaps and reassemble the original order. It had worked on a bacterium with under 2 million letters. Human DNA was 1,000 times larger. The objection was repetition. Large stretches of human chromosomes consist of sequences repeated over and over, sometimes for hundreds of thousands of letters. A computer trying to place a fragment inside such a region has no unique landmark to anchor it. Critics said the assembly would collapse into ambiguity. Celera built one of the largest civilian computing installations in the world to attempt it, and bought hundreds of capillary sequencers. The public consortium responded by abandoning its original timetable. Instead of finishing chromosome by chromosome, it would produce a rough draft of everything, fast, and polish later. The rivalry was real and often unpleasant. Venter argued the public effort was slow and bureaucratic. Consortium scientists argued that Celera was using freely released public data to scaffold its own assembly while planning to sell subscription access to its results. Both accusations contained some truth. Pressure came from outside science as well. Investors watched. Governments watched. In March 2000, a joint statement by President Bill Clinton and Prime Minister Tony Blair urging that raw human sequence data remain freely available knocked billions of dollars off the value of biotechnology stocks within days. On June 26, 2000, the two sides appeared together in the East Room of the White House. Clinton stood between Collins and Venter and announced that a working draft of the human genome was complete. The negotiated truce had been brokered largely by Ari Petrinos of the Department of Energy, over dinners at his home. The draft covered roughly 90% of the sequence, with thousands of gaps and an error rate that made many regions provisional. It was an announcement of arrival, not of completion. Both groups then spent 7 more months checking, arguing and writing up what they had actually found in the sequence. The papers appeared in February 2001. The consortium published in Nature, Celera in Science. Together they described about 3.1 billion base pairs of human DNA, the 4 chemical letters that spell out a person, laid end to end in order for the first time. Then came the number nobody expected. Before sequencing, estimates of how many protein-coding genes a human carries ranged from about 50,000 to well over 100,000. There had even been an informal betting pool running among researchers at scientific meetings, and most of the wagers were placed high. The draft suggested somewhere between 30 and 40,000. Refinement pushed it down further, and the figure settled at roughly 20,000 protein-coding genes. That is close to the count in a microscopic roundworm, and fewer than in many plants. A grain of rice carries more. It reframed the entire question. Complexity in humans does not come from owning more genes. It comes from how those genes are regulated, spliced into multiple products, switched on in particular cells at particular moments. Most of the genome does not code for protein at all, and much of it does other work entirely. Roughly half of human DNA turned out to be repeated elements, including remnants of ancient viruses that inserted themselves into the germline of our ancestors and stayed. Some are inert. Some have been recruited to do useful jobs. The genome reads less like a blueprint and more like an archive. On April 14, 2003, the consortium declared the project complete, 50 years after the description of the double helix and 2 years ahead of the original schedule. The final public cost is usually given as roughly $2.7 billion, less than the $3 billion originally budgeted. Complete, in that announcement, meant something specific. It meant about 92% of the genome, covering essentially all the gene-rich regions, at high accuracy. The remaining 8% was not forgotten. It was simply unreadable with the sequencing chemistry available at the time, and everyone involved knew which parts were missing. Those regions were the repetitive ones. Centromeres, the dense knots at the middle of each chromosome. The short arms of 5 chromosomes. Long tandem arrays where the same motif recurs thousands of times. Sanger sequencing produced fragments too short to span them, so the gap stayed open for 19 more years. New instruments eventually solved it. Long-read sequencers, which thread a single DNA molecule through a tiny pore or watch a single enzyme copy it in real time, can produce reads hundreds of thousands of letters long. That is enough to cross a repeat and land on solid ground on the far side. In 2022, the Telomere-to-Telomere Consortium published the first gapless human genome sequence, adding almost 200 million base pairs that had never been read. It was assembled largely from a cell line with an unusual genetic makeup that simplified the problem, which made the work possible. Cost has fallen further than almost anyone predicted. The first genome took 13 years and billions of dollars. Sequencing a human genome today is routinely quoted in the hundreds of dollars and takes about a day. The instrument that does it fits on a laboratory bench. The medical payoff arrived more slowly than the 1990s promised. Cures for common diseases did not follow automatically from having the sequence. What did follow was diagnosis. Children with rare undiagnosed conditions can now have their genomes read and an answer found in weeks rather than years of testing. Cancer treatment changed too. Tumors are sequenced to find the specific mutations driving them, and drugs are chosen accordingly. Population studies comparing millions of genomes have identified thousands of variants that shift disease risk, most by small amounts, which is itself a finding about how biology works. The project also left behind something less tangible. The Bermuda Principles established that a fundamental reference for all of human biology should be public property, released daily, free to use. That decision, made in a hotel meeting in 1996, shaped how genomic data has been shared ever since. 13 years of work produced a sequence, and the sequence produced a smaller number than anyone wanted. 20,000 genes to build a human being. The reference that took a consortium and a rival company more than a decade to assemble can now be read overnight, and the hard part was never the reading.